Hi, good morning, everybody. I'm Taiana Maia De Oliveira from AstraZeneca. I'm here to present the talk entitled Cryo-EM Reviews Structural Insights on DNA Damage Repair. So this talk will actually focus on the findings on 3 proteins which are at the heart of DNA damage repair. These are ATM, ATR, and DNAPKcs. These studies were the product of 2 collaborations with 2 incredibly talented groups in Cambridge, the Roger Williams group from the LMB and the Tom Blundell group from the University of Cambridge. So each cell in the human body receives tens of thousands of DNA lesions per day. DNA damage is detected and repaired by DNA repair pathways like the ones illustrated in this slide here. And as I said before, this talk will focus on these 3 proteins, ATM, ATR, and DNAPKcs. So let's go a bit through the basics of what these proteins actually do. So ATM is important for the repair of DNA double-stranded breaks. Let's imagine that we have a DNA double-stranded break here, and that the cell is in S phase or in G2 phase in which a sister chromatid is available. What will happen next is that the mRNA complex will locate to the break. Once there, this complex will be able to recruit ATM, which will become active. Active ATM will then phosphorylate several substrates, promoting the accumulation of homologous recombination factors that will promote the repair. Now let's focus a bit on ATR. So ATR can be activated by a variety of damages, like for example, nicks or defective replication forks. These defects are then coated with a protein named RPA, and RPA is then able to recruit ATR via its obligatory partner ATRIP. Active ATR is then able to phosphorylate several factors, like for example, CHEK1, and these factors will promote checkpoint activation, fork stabilization, and finally repair. Last but not least, we'll go to DNAPKcs. So DNAPKcs is also important for the repair of DNA double-stranded break, but in a case in which the sister chromatid is not available. That means that the cell will then take the pathway of non-homologous end joining here. What will happen in this case is that Q70 and Q80 will locate into the DNA break, and then this will trigger the recruitment of DNAPKcs. The Q heterodimer together with DNAPKcs forms a complex called DNAPK. Active DNAPK will phosphorylate several substrates, and these will accumulate on the break. And these include, for example, XLF and XRCC4. And all these factors will act together to finally make possible the ligation of the ends by ligase 4. So you might ask now why AstraZeneca is interested in these proteins. And this is mainly because they have been linked to tumorigenesis and survival of cancer cells after therapy. Because of that fact, DNA damage response is actually one of AstraZeneca's areas of focus within oncology. So interestingly, these 3 proteins belong to the family of the phosphoinositol 3 kinase related kinases, the PIKKs. These kinases, they're structurally related to the lipid kinase PI3K, but they don't phosphorylate lipids, they phosphorylate protein substrates. Other famous members of this family is the cell growth regulator mTOR. So given the importance of these proteins, you can imagine that the pursuit of 3D structures was intensive for very many years, but these efforts found very little success. That is until the Cryo-EM revolution came, and that finally allowed us to really see these spectacular molecular machines for the first time. All these EM structures that came from that, they confirmed that these proteins share a similar domain organization. I will elaborate a little bit about this organization because they are going to be referred to later in the talk. So these proteins, they feature an alpha helical and terminus here, which is the solenoid. It's made of heat repeats. This is followed by another helical domain, which is called the fat domain. This is followed by the kinase domain, and finally by a small domain, C-terminal to the fat C domain. One of the features of these kinases is that they have a canonical kinase domain, but this domain is very intimately connected to the fat and the fat C domain, and they form one structural element, which is the fatkin. Another particular feature of these kinases is the fact that they can have insertions in the kinase domain. One of them is the famous insertion, the FRB domain, and another one is one that we'll refer to later, the PIKK regulatory domain, which we'll explore a bit on the other slides. So let's start with the cryo-EM structure of ATM. This was a work that was mainly driven by Dr. Dome Baretic, who was a very talented AstraZeneca LMB Blue Skies postdoc at the Williams Lab. So AstraZeneca had established a protocol to express small quantities of very high quality ATM in mammalian cells. The EM analysis of the sample revealed that the ATM existed in an equilibrium between a closed symmetric dimer and an open asymmetric dimer. And that right away prompted us to question what was the physiological relevance of these different forms. Now, to answer that question, we'll have to look a bit closer at this one structural element that I talked about, the fatkin. So in the case of the closed symmetric dimer, which is here, I would like to draw your attention to these helixes here in purple. You can see that these helixes that belong to this one protomer here are binding to the helixes in cyan, red, and green from the next protomer. So in fact, these purple helixes are pushing this structural element into the substrate site of the kinase. That's something that you can appreciate by this close-up here. This is then an auto-inhibited state of ATM. Now we can look a bit at the asymmetric dimer. So the first protomer here is rotated about 25 degrees, and that in fact moves the purple helixes away and release the structural element from the substrate site. So this conformation of ATM is not auto-inhibited. The substrate site is available. Now, something that is curious here is actually that these green helixes here, they are part of the domain that I told you about in the introduction, the PIKK regulatory domain, which I'll now refer to as PRD. These structures were very interesting because they explain years of biochemistry studies that reported that mutations on the PRD had profound effects on the activities of these kinases. So now let's go to the cryo-EM structure of ATR, which was also mostly driven by Domi. So for ATR, like for ATM, we had two different populations of particles observed in the sample. The first corresponds to a symmetric dimer and is similar to the one that was published by another group in a paper by Lau and co-authors in 2018. This structure, our structure, actually happened to have a better resolution in the ATRIP part, and that allows us to clarify a discussion point of the Lau paper regarding the stoichiometry of the complex. In our structure, it was clear that we actually had two ATRs interacting with two ATRIPs in the complex. But going back into the two conformations that we see in the sample, we have a compact conformation. That's the one that is similar to the one that has been published. And this is the another extra conformation, which we call as the extended conformation. And I think from this figure here, you can appreciate that there are actually quite some differences on the solenoid of ATR and also on the ATRIP part. If we look at this view here from the bottom, you can see that in the compact conformation, there's a contact between the two ATRIP molecules, and that this contact is actually disrupted on the extended conformation. Now, because there are biochemistry studies reporting that ATRIP dimerization is essential for ATR activity, we can speculate that this extended conformation could be relevant for a kind of downregulation of ATR activity in the cell in a certain context. Now, in this final part, I will talk about the recent work which came from a collaboration with Sir Tom Blundell and his group. This was work mostly driven by Dr. Amanda Chaplin, who was a postdoc at Tom's lab and is now just accepted a position at the University of Leicester as a lecturer. So a little bit, I'll give a little bit of context here. The group of Sir Tom Blundell has been working with these DNAPK structures for a long time. The first structures that came from his lab were obtained by absolutely titanic efforts in X-ray crystallographies, in which they tested thousands of crystals until getting something that diffracted. With Cryo-EM, high resolution structure of DNAPKcs in complex of CUT70 and 80 and DNA could be obtained in much less time. So this first slide is referring to that structure. This map allowed us for the first time to really model the kinase in an ambiguous way. What was interesting in this sample is that it seems to be a repeating theme here. There were also other different conformations on that. And we could actually see a dimer that was mediated by the C-terminus of Q80. This is this figure here, illustrating the Q80 in green. So this dimer could be one of the possible synapses holding the DNA ends at a good proximity to allow for repair. This is supported by biochemical observations that cells that are lacking the C-terminus of Q80 have very low DNAPK activity, they are radiation sensitive, and they are also unable to conduct proper VDJ recombination, which is one of the roles of non-homologous end joining in the cell. Now, one of our ambitions in applying Cryo-EM to the study of these non-homologous end joining complexes was really to visualize the entire pathway. You can imagine all these textbook figures in which you have all the proteins represented as circles, triangles, and this, and then you have the break and everything accumulating. And we really would like to replace that figure for something that is more realistic and that really shows the mechanism of action of each individual cell. So we tried putting all of this together. So in this sample, we have DNA, DNAPKcs, CUT70, Q80, XLF, XRCC4, and ligase 4. We could actually see a super complex of non-homologous end joining in the sample. And surprisingly, this indicated yet a new dimer. This is a new dimer that is mediated by XLF instead of being mediated by Q80 like the previous one that was shown. And one of the remarkable facts about these dimers is there's actually a lot of interactions between the DNAPK-CS solenoids, and this seems to be the interface that is mostly relevant to keep this dimer together. Mutations of the interfaces of both this dimer and the dimer that I showed in the previous slide, they prevent recombination in cell. So we think that this dimer is another of the possible synapses that hold the DNA ends in a good position to allow for repair. Now, this slide here is to illustrate the distance between the DNA ends in the XLF-mediated dimer here and the co-mediated dimer here. So you can see that in both of these structures, the distance between the DNA ends is approximately 115 angstrom. This distance is consistent with something that has been reported in the literature, which is an experiment of single molecule FRET that described long-range synaptic assemblies having this distance between the DNA ends. So finally, I think these structures, together with another structure that has been published by a different group, this is a paper by Chen et al. From this year in Nature, showing a similar supercomplex to the one that we have here, but lacking DNAPK-CS on it. They allow for a possible interpretation of the overall mechanism of non-homologous end joining. So in this mechanism, we think that the co-heterodimer is coming, localizing to the DNA ends, recruiting DNAPK-CS, and then in that case, DNAPK-CS and DNAPK will be found in one of the possible complexes, which are the ones that we seen in both papers. So they would organize in either the first dimer that is mediated by Q80 or the dimer mediated by XLF. Now, these would be the long-range synaptic complexes. What could happen next is that DNAPK would be phosphorylated and eventually it would be removed. That would trigger the formation of the short-range synaptic complex, which could then bring the DNA ends closer together and in a position to actually be ligated by ligase 4. So finally, I would like to thank all the marvelous collaborators, especially the ones that from the Williams group and from the Blundell's group who have enabled all of this. I feel very privileged to be able to work with this incredible pool of very talented scientists. So thank you very much. And of course, I would like to thank the organizers for the opportunity to talk. And finally, I would like to thank you all for the attention, and I would be happy to take any questions if there are any.
Well, thanks very much, Taiana. I'll go over to Chris and see if there's any questions. Well, I think everybody's getting kind of maybe in need of some coffee. So I mean, I'd kind of like to ask you, I mean, how difficult was it to actually get these complexes then into the cryo-EM stage? What was this kind of the stumbling blocks?
No, it was a lot of trial and error, I think, as everything else in cryo-EM. Putting proteins in the different, I think, especially for the non-homologous end joining, is really trying with different ratios of protein, with different sizes of DNA, and really collecting data and seeing if that would give something that was suitable for further analysis or not.
And are you going to go further on with these three structures and then look at, say, like the regulation in terms of phosphorylation or other modifications?
Yeah, so we have a little bit of a hint already in one of these structures. Let me see if I can go here. In the in the dimer that is mediated by XLF, actually, two of the main parts of DNAPK that are very important phosphorylation, which are the PQR and the ABCDE, are in close proximity to the kinase domain in this dimer. So it could be that this is actually the position for transphosphorylation. However, that there are reports of cis phosphorylation too. So it's we don't know yet if this is the main state of the kinase that would be responsible for the phosphorylation.